short. I know the coffee break is beckoning. I'm going to finish off this part of the session by moving a little bit sideways into the more behavioural aspects of feed efficiency. This work was carried out with colleagues from AFBI here in Northern Ireland, VetMed Uni, Chuggas in the Republic, and Queen's here in Belfast. You're all by now very familiar with the problems that we face in terms of an increasing global population, the need for sustainable intensification of agriculture, and the role that feed efficiency can play in this. As you've been hearing, there are so many possible causes of variation in feed efficiency, and including things that we've been hearing about, such as the gut microbiome, gut structure and function, and to this we can add feeding behaviour. Feeding behaviour can affect both the digestive process and the amount of energy that an animal uses in its daily life, for example, in trips to and from a feeder. We know that differences in feeding behaviour have been found between animals of different breeds and different lines, which differ in feed efficiency. But we need to look more closely within populations to see what effects the feeding behaviour of very similar animals can have on their feed efficiency. So the aim of this work was to investigate relationships between feed use efficiency and feeding behaviour in pigs housed in entire litter groups. And this work was done using data from a study that Tanya described at the very start of this session. So briefly, the work was carried out on 3 different sites: the Republic of Ireland, Northern Ireland, and Austria. The animals on the different sites had similar genetics, and common boards as well as site-specific boards were used. The feed was formulated the same on the 3 sites, and management was kept as similar as possible. There were some environmental differences, for example, the season in which studies were carried out, and also photo period due to geographical differences. So there were 4 batches of pigs on the 3 sites, 2 batches in Chuggas in the Republic of Ireland, and 1 batch at each of the other sites. The pigs were weaned at 28 days, and in the finishing period, the period that we were interested in for this work, they were housed in pens of 7 to 13 litter mates. The pigs were fed ad lib through automatic single-space feeders, 1 feeder per pen, and these feeders recorded all of the feeding data for each of the animals continuously, plus or minus a few technical difficulties. We then selected intake data for 2 24-hour periods per week in weeks 12 to 20 of life. These 2 weekly periods were averaged to give an average value for each week, and then these weekly averages were overall averaged again to give 1 overall average value per pig for weeks 12 to 20. And these parameters were things like total feed intake, duration of feeding, number of feeding bouts, and the size and duration of the feeding bouts. Feed efficiency was measured using RFI, which you've heard a lot about already today. There were also some other things that we wanted to account for in the analysis. These pigs were sharing pens and they were sharing feeders. So there are a number of things that can affect the feeding behaviour, including the gender of the pigs and also the weight of the pigs compared to their litter mates. These things have been shown in previous studies. So for the weight of the pigs, we ranked the pigs in each pen in relation to their relative weight. So a group of heaviest, medium weight and lightest weight pigs, and then we were able to include this in the analysis. So data was analysed using a Remmel linear mixed model in GenStat. The fixed model included RFI, gender and relative weight, and the random model included litter within batch and bore. And 4 of the parameters were log transformed, and I'll point these out as we go on to improve the normality of the residuals. There were only 2 results tables in this whole presentation. This first table shows the overall parameters for feeding behaviour. So this first column is the median values. The second is the coefficient for RFI from the linear model. And the third column is the significance of the relationship between RFI and the feeding behaviour parameter. So for the total feed intake in 24 hours, a median value of 2.4 kilos, a positive coefficient and a significant relationship between RFI and total feed intake, as you would expect. For total time feeding in 24 hours, a median of around 66 minutes. Again, a positive coefficient and a significant relationship between RFI and total duration. And then for the rate of feed intake, a median of around 0.6 grams per second, a positive coefficient and a significant relationship between rate of feeding and RFI. So when we remember that higher RFI equates to lower feed efficiency, these less efficient pigs had a higher total intake and a longer total time feeding, and higher RFI was also associated with a faster rate of feeding. To move on to the results for the individual feeding bouts or visits to the feeder, these are the parameters that were log transformed. The number of feeding bouts per 24 hours, a median of around 15 bouts, a positive coefficient and a significant relationship between RFI and the number of feeding bouts. When we look at the number of feeding bouts per kilogram of feed eaten, the median then is 6.5 roughly. The coefficient wasn't different from 0, and there was no significant relationship between RFI and the number of bouts per kilo. Size of feeding bouts, median 170 grams. Again, coefficient not different from 0, and no significant relationship. And feeding bout duration, median of around 4.7 minutes. Coefficient not different from 0, and again, no significant relationship. So from these results, the less efficient pigs had more feeding bouts in 24 hours, but not more bouts per kilo of feed consumed. And there was no difference in bout size or bout duration. So discussion, in terms of overall feed intake and feeding activity, as you would expect, the less efficient pigs had a higher intake, and associated with this, a longer duration of feeding and a higher number of bouts. Now, although they had a higher number of bouts in total, when you divided this by intake to get the number of bouts per kilo of feed consumed, there wasn't a difference. And there was also no difference in the characteristics of the bouts, so the size or the duration. So it doesn't seem that the less efficient animals are spending a lot of time walking to and from the feeder, having many tiny bouts. The main difference seems to be in the amount that they're eating in total. The results in terms of feeding rate are interesting. Now, a higher feeding rate hasn't previously been associated with a poor feed efficiency. And in fact, a previous study, in 2010, found that when they compared two lines that had been selected on the basis of feed efficiency, that actually the more efficient line had a faster rate of feeding. When they then looked within this line, though, they found that the relationship wasn't there. So it seems that there is possibly quite a lot of variation, again, as we've been hearing all afternoon, even between lines, between breeds and between populations. The designs of many of these experiments looking at feeding behaviour and feed efficiency, they're all very different, and the analyses are different again. The experimental design of this study allowed the environment to be kept constant within litters, and we were able to include the relative weight of each animal and gender in the model, which might have allowed us to see things that haven't been clear in previous work. But if this relationship does exist, how could feeding rate affect RFI? Well, intuitively, feeding more slowly might allow more therapeutic, better mixing of the feed with saliva, and smaller particle size entering the digestive system. It might also allow more contact with digestive enzymes and the absorptive surface. And there's been some very interesting research in humans looking at the feeding rate and total intake of people. So in humans, a higher feeding rate is associated with a higher total intake. And if you experimentally ask people to reduce their feeding rate, they will actually reduce their intake without knowing it. So the implications of this, well, as we have been hearing repeatedly, the relationships between RFI and physical and environmental factors are very complicated, and do seem to vary by environment and also by breed and strain. Even within this study, we did find some batch differences. When we looked at the relationship between feeding rate and RFI, it wasn't identical at all of the sites. But if this was a widespread relationship, might there be scope to improve RFI by manipulating feeding rate? It's possible in some more highly automated systems, possibly, to identify animals which have a higher than expected intake and to reduce specifically the rate of those animals. For example, by changing the speed at which feed drops from the hopper into the feeding trough in an automatic feeder. But any manipulation that you make to change the rate of feeding could have knock-on effects on other aspects of productivity, so it would have to be very carefully researched and balanced. We still are working on this data set. There are lots more very interesting questions to ask. We're looking at links between feeding behaviour and the weight of the digestive tract, the different digestive organs. We're examining the circadian pattern of feeding, and some interesting links that seem to be there with RFI, and also looking at relationships between those feeding behaviour parameters, live weight and back fat.